Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Oleksandr. Let's start. Three, two, one, and we are live. Welcome to the next episode of the Personalized Medicine Podcast. Back in 2019, in episode 3, we have discussed glycans, a very important and somewhat neglected group of biomarkers. Recently, glycans have been receiving a lot of attention. It turns out the surface of coronavirus is packed with glycans, which help this virus to evade our immune system. The recent research of our today's guest sheds the light on the structure of that core surface glycoprotein of coronavirus, and hence can facilitate the development of vaccine against it. I'm very happy to introduce today on our show Dr. Parastoo Azadi. Parastoo is the Technical Director of Analytical Services at Complex Carbohydrate Research Center in the US. She received her bachelor's in chemistry from University of North London and her PhD degree in biochemistry from Imperial College of Science and Technology, where she studied structural characterization of carbohydrates and glycoproteins by mass spectrometry. She then joined MScan Limited, an analytical mass spectrometry consultancy where she was a study director responsible for complete structural characterization of native and recombinant proteins and glycoproteins using mass spectrometry. In 1994, Parastoo joined the Complex Carbohydrate Research Center at University of Georgia. Since 2001, she has been working as a technical director at the center, with her research focusing on characterization of glycoproteins. She has more than 130 papers under her belt, and most recently her group has published 2 very exciting publications on coronavirus. So, Parastoo, welcome on the podcast. It's a real pleasure to welcome you today on our show.
Thank you. Thank you for a wonderful introduction. I'm happy to be here today.
Perfect. I would like to start with one of your most recent research papers in Glycobiology journal, in which you investigate glycosylation of coronavirus spike protein. Could you briefly summarize the main findings of the study and what do they mean for our fight against COVID pandemic?
So the coronavirus SARS-CoV-2 has a glycoprotein on its surface called the spike protein. This protein covers all the surface of the virus, and it's important for its entry into the host cells, which into humans. We decided to look at the structure of the glycans on the surface of the coronavirus and to elucidate the site of glycosylation and the glycan structures in order to determine maybe it can help in figuring out how this coronavirus gets into the host cell, how it gets attached to the host cells. So we looked at this glycoprotein, which is made up of 22 different glycosylation sites. The spike protein is made up of 2 subunits, S1 and S2. We looked at these S1 and S2 subunits independently because we think that probably for vaccine development, maybe not the whole glycoproteins may be used for vaccine, maybe only the S1 subunit, which has the binding region, that the region that binds to the host cell may be only used for vaccine. So we looked at the different subunits independently to know the glycosylation and how this may affect binding. So we found out that this spike protein has 22 glycosylation sites, and out of these 22 glycosylation sites, 17 of those is glycosylated. Interestingly, we found 2 O-glycosylation sites. One has been confirmed unambiguously. One probably needs more work to confirm that it's definitely glycosylated or not. But these O-glycosylation sites could be critical for binding of the spike protein to the host cells because these 2 O-glycosylation sites are very close to the binding region. and they couldn't affect on the way the protein binds to the receptor. Also, there are other regions of the glycoprotein that could be important for binding, and the type of glycans present on there could determine the type of binding there is to the receptor. There are different glycans, for example, the high mannose glycans, basically complex type glycans, silylated glycans, all these different heterogeneities within the glycoproteins could influence binding. So it's important to know the whole glycan structures in order to develop a vaccine, whatever platform that may be, using the spike protein.
Yeah, I guess what you just mentioned is really important. The differential glycosylation of that spike protein can perhaps explain why infectivity of this virus differs between different strains. Do you have any data to support or disregard that hypothesis right now?
At the moment, we don't have any direct evidence for which glycans are contributing to any differential infections in coronavirus, or which glycan structures are doing that. But there are mutants in coronavirus, and it will be important to study these further to find out these mutations, are they the mutations in the sequence of the different spike protein, for example? Are there also differences in glycosylation? What I mean is the differences in the amino acids. If they're close to the glycosylation sites, or they are including some glycosylation site regions, are they contributing to the heterogeneity, and are they contributing to the different infections of the virus?
Perfect. Understood. And I know that apart from studying the spike protein of coronavirus, you also studied the surface receptor on the human cells, the ACE2, that is responsible for interaction with that spike protein. And from what I understand, it's also a glycoprotein. Can you tell us a little bit about that line of your recent research?
Yes, so the coronavirus is believed to bind to this angiotensin receptor, ACE2 receptor, and probably is not the only receptor, but it's been shown that it's one of the main receptors that this spike protein binds to. So there was no other previous comprehensive glycosylation study on this protein, although the structure has been known since 2000. It has 7 glycosylation sites, and we've shown that all the sites are glycosylated. Interestingly, we found 3 O-glycosylation sites in this protein that has not been reported before. It has very interesting glycosylation profile. There is 85% fucosylation, there is 50% silylation, and maybe more importantly, is the sialic acid linkages that we determined that the 2,3 link sialic acid is the major sialic acid that we've observed in ACE2, and none of the glycans are just only 2,6 linked. Sialic acids has been shown to be important for viral infections previously in other viruses, so it's interesting to see whether the linkages of the glycans on ACE2, how relevant these will be for the way the spike attaches to ACE2. So, for example, the N-terminal domain of MERS-CoV protein prefers 2,3 link sialic acids over 2,6, and we found that the 2,3 link sialic acid is the main linkage of sialic acids in ACE2. So it will be interesting to find out the importance of these linkages and their implications for the binding of the spike to ACE2.
Perfect, understood. So essentially, that can potentially mean that can potentially explain why we have this different response of different people to coronavirus. Some seem to be not affected as much as others, and perhaps part of the answer can be in that differential glycosylation profile of the surface receptors that are responsible for interaction with the virus itself.
Yes, I mean, it could be that is the glycosylation differences between different people who's contributing to the differences that people are reacting to the coronavirus. It's the glycosylation profiles on their ACE2 or other receptors that are there. ACE2 could be just one of the receptors. The host of receptors that are there and their glycosylation profiles differences could contribute to the way spike is attaching to these receptors.
Understood. Perfect. And I guess one more topic that I wanted to discuss with you around the coronavirus is why is it so difficult to actually develop the vaccine against those glycoproteins or viruses that are covered with glycans?
So for SARS-CoV-2, it's challenging because it's a new virus for us. So scientists don't know anything about it. Our immune system doesn't know anything about it. So it's going to be challenging. We take cues from other coronaviruses, other virus research that has been going on. But it is challenging because it's all new. That's the first thing I want to say. The second thing is that we don't have any previous clinical trials. We don't have any. There's no medications. There's no other vaccines that it's shown to work even partially for this coronavirus. The spike protein is particularly complicated, particularly heterogeneous. Having 22 glycosylation sites and 17 of those, or maybe most of those being glycosylated and heavily glycosylated with very heterogeneous profile makes it challenging to develop vaccines against this virus. So basically, a vaccine triggers a response, and we want to be able to capture that response safely and effectively, so that it can protect us against the infection. The whole aim of a vaccine is to produce a robust antibody response with neutralizing antibodies, coupled with some sort of cellular response to make sure that it's prolonged and we get immunity from this kind of response in our body.
Makes perfect sense. And these are the challenges that scientific community is facing now with coronavirus that are not so easy to overcome to generate an effective vaccine. So beside coronavirus, I know that you have done a lot of exciting research, both with academic and industrial collaborators over the years. What were 2 or 3 highlights in your research career that were really the most exciting kind of topics that you had chance to work with so far?
Over the last 30 years, I've worked with government agencies and research institutes and pharmaceutical industry. We've done a lot of work, internal research on developing tools for glycoprotein and polysaccharide analysis. We've been very fortunate to have funding from Department of Energy over the years in being a resource to the glycoscience community in developing tools for polysaccharide, plant and microbial polysaccharide analysis. We currently have a large center grant concentrating on looking at methods for structural characterization of plant polysaccharides, especially specific polysaccharides in the plant cell wall. So that's an exciting area in developing tools that will be important for looking at plant polysaccharides and its implications for biofuel. Of course, I've worked with a lot of industry, and a lot of work in the last 10 years of my lab has been for looking at structural characterization of heparin and low molecular weight heparin products. As you may know, in 2008, there was a contamination in heparin, and we and others quickly started working on developing new techniques to be able to capture the full structure of heparin products and to be able to tell quickly if these products are contaminated with any, like in that case, it was oversulfated chondroitin sulfate. So we have NMR tools and mass spec tools that can really elucidate fine structure of these very complicated polysaccharide mixtures and their sulfation patterns and their sequence, and basically in a mixture.
Perfect, understood. And I think that just shows like the breadth of importance of glycans in biology and in technology, because starting with the biofuel and ended up with the quality control for therapeutic products, they are essentially all over these fields. And it's important to have reliable, robust tools to be able to assess those glycans and their role in all those processes. We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview. So you mentioned actually that you've been working in this field for already 30 years. And for your PhD already, very early in the beginning, you were using mass spectrometry to study those glycans. Even today, I think it is still quite challenging to study glycoproteins using mass spectrometry. But I can only imagine how it was back then, 30 years ago. So maybe you can share with our audience a little bit what was actually possible to do with mass spectrometry back at that time, and how tedious was it to set up an experiment in the lab to study glycans with mass spectrometry 30 years ago?
Sure, that's interesting. I'm dating myself. So when I joined the lab of Andel and Howard Morris at Imperial College to study mass spectrometry of glycans, the instrument of choice at the time was FAB-MS, fast atom bombardment mass spectrometry. And actually my PhD was sponsored by VG Analytical at the time, that they were making the instruments. So they wanted to have students who are familiar with the FAB-MS techniques and looking at biopolymers. So our lab was developing techniques for looking at biopolymers as a whole, proteins and carbohydrates. So FAB-MS was basically a technique that was an ionization technique that had a magnetic sector instrument attached to it. And magnetic sector instruments as a whole were a different ballgame than the MOLD DMS or the electrospray that we use nowadays. They actually, at the time, they were extremely high resolution mass spec for its time, because it allowed high resolution mass spec at the same time being able to elucidate the structure. So it allowed us to elucidate the structure of the glycans and proteins, peptides accurately. But they were difficult to use. First of all, these took a long time to be able to calibrate these instruments. It's not like right now that the MOLDI, you basically push a button and it gets calibrated, or electrospray, you push a button, it gets calibrated. It took maybe an hour, sometimes an hour and a half, depending on which instruments we use, to properly calibrate it. And nowadays, most instruments, you don't have to calibrate them even once a week or maybe even 2 weeks or something like that, electrospace or MALDIs. But then for fast atom bombardments, we had to calibrate it every day. We were able to get very accurate peptide mapping and glycan mapping data for a number of glycoproteins. And then in the late 80s, the electrospray technique was developed by Fenn and his groups. And actually, I analyzed the first glycoprotein on electrospray, which was a C-reactive glycoprotein using intact mass analysis of the first glycoprotein using electrospray in the late 80s when I was still at Imperial College.
Great. Yeah, that sounds very exciting. And of course, like the technology was very different, but still a lot was possible even back then. And I think FabMS, probably a lot of our listeners who are even familiar with mass spectrometry will not have never used it and probably will not use it.
Probably I've never seen it.
Yeah, exactly. So maybe we'll try to find a picture of the Fab mass spectrometer and we'll put it to the show notes so our most passionate mass spec listeners can have a look. How has it looked? Perfect. It sounds really exciting and also like shows the dedication, like how much had to be done essentially to perform an experiment that today would be much simpler. And also it's a good sign because it shows how far the technology has progressed over the last years. You also mentioned that at your institute, you not just use mass spectrometry to study glycans, but also use NMR, nuclear magnetic resonance. Can you tell our audience a little bit about when would be a good time to use NMR? Like what would be the reason to use NMR to study glycans or glycoproteins? And what are the key advantages of this method, for example, over mass spectrometry?
Sure. Glycans are complicated structures, so they are not linear, they're branched, so we will need to know the sequence of these glycans, we need to know the linkages. So only mass spectrometry is not going to be, only mass spectrometry will not give you the whole polysaccharide or glycan sequence. So we will need complementary techniques. We use NMR, we use GCMS. We use GCMS quite a lot, actually, for linkage analysis. So NMR is used if, for example, we have a polysaccharide and we need to know the sequence. Mass spec is not as useful for looking at the structure of the polysaccharides because mass spec doesn't have the mass range that we need for polysaccharide analysis unless we digest it or cut it into smaller parts. But if we want to look at the intact polysaccharide or intact glycan, and we don't want to digest them in any way, we can look at NMR, and NMR is able to give you the sequence, the linkage, the alpha-beta configurations, all the information you need for basically glycoconjugate characterization as a whole. The disadvantages for NMR analysis, samples needs to be fairly homogeneous. So we cannot look at big mixtures usually by NMR. You can look at big mixtures by NMR, but you get some kind of a profiling of everything. You cannot sequence everything. In order to sequence polysaccharides, they need to be homogeneous. So we do quite a bit of purification and basically isolation of lipopolysaccharides and exopolysaccharides, capsular polysaccharides, for example, or plant polysaccharides prior to NMR analysis. So we figure out if experiment can be done by NMR first if it's homogeneous. If not, then we will purify it prior to it doing NMR.
Perfect, understood. So each method has its advantages and disadvantages, and I guess the carbohydrates, polysaccharides are so complex that it's really hard to use just a single method to understand both sequence and structure of that glycan, especially in the complex mixture. So my next question is also technical, and it's actually more on the deciphering those oligosaccharide trees on the surface of proteins, because as you said, individual monosaccharides can be linked to each other in a very different manner, and that would ultimately determine the structure and the shape of this oligosaccharide tree. So how do you combine mass spectrometry with some other techniques to decipher the sequence and those linkages of individual sugar molecules in that oligosaccharide tree?
So we use a combination of techniques, as you said, in order to get the full picture of what the glycoprotein structure is or what the polysaccharide structure is. So if we are talking about a glycoprotein, we need to know what are the glycans attached, but we also need to know what are the sites of attachments. So we do some kind of a peptide mapping, glycopeptide mapping, to look at the sites of glycosylation. So we figure out what is the peptide backbone, and if that site, for example, asparagine or threonine residue is glycosylated, then we use the mass spec to basically sequence the glycan. We usually release the glycans and derivatize them using permethylation or acetylation to basically tell us what is happening at the reducing end, what are the glycan structures at the non-reducing end, and for example, the ratio of fucose being at one end of the molecule versus fucose being on the other end. What are the sialic acids? What is the position of the sialic acids? These fine-tuning of the glycan structures is done through derivatization and then basically tandem mass spectrometry. Different tandem mass spectrometry techniques or hybrid fragmentation methods are used for elucidating the full structure of the glycans. But sometimes just the mass spec of just using maybe electrospray or MALDI is not enough. So we look into GCMS. We make partially methylated alditol acetates, and then we look at the linkage positions of these monosaccharides that are attached to each other. For example, the linkage of the galactose, the linkage of the glucnac or the galnac that is present, the linkages of the fucose. So combination of the derivatization using GCMS and LCMS and electrospray using MSN or fragmentations can help give you the full structure of the glycoprotein.
Perfect. Yeah, that sounds complex, but I guess so is the biology of glycoproteins. So we need to use those complicated methods, sometimes complicated methods, to fully understand how those building blocks of our body are built.
But one important thing that I think I'd like to also mention is I think so far we've talked about purified glycoproteins, but it's important to be able to look at glycoproteins on mixtures, on cell surfaces, on different cell lines, for example, or different tissues. So those looking at real research comes into being able to look at mixture of the glycoproteins together. So, for example, looking at cancer cells versus normal cells, different tissues. So the challenge is to be able to observe those glycans accurately and to be able to capture those glycan signature glycan structures accurately and sensitively in order to know what are the differences between these tissues, what are the biomarkers in these tissues.
Absolutely, and I think that's really a prerequisite for successful personalized medicine, when we could essentially detect specific proteins or glycoproteins in those complex mixtures coming from either blood serum or tissue, for example, from cancer biopsies, as you just mentioned. And that could really help us stratify patients, understand which therapeutics to use, and how well can they respond to specific treatments.
Yeah, basically, you're looking at real-time glycosylation changes is important. And for those, it's very challenging because the glycan signals usually are much reduced compared to peptide signals. So we need to have a way of enriching those glycan signals, those glycopeptide signals, in order to bring them to the surface and in order for us to see, for them to be highlighted rather than being buried with every other signal. So now we and a lot of other people are developing techniques to be able to enrich for these glycan structures and enhance their sensitivity.
Right. And what those techniques would be, is there something like a classical immunoprecipitation using specific antibodies against glycosylated proteins, or there are some other more specific techniques that can be used to enrich those glycoproteins in the sample?
So people are using antibodies, they're using a lot of lectins to basically enrich for a particular structure, or using different columns that are more specific. They will bind specifically to more glycans, for example, Hilli columns maybe, to basically enrich the glycopeptide or glycoproteins prior to analysis by mass spec or other HPLC techniques, maybe using fluorescent detections, because sometimes the changes in glycosylation are very subtle, and maybe we will need some kind of fluorescent detections, tagging and fluorescent detections in order to detect these subtle changes in glycosylations. And it's not just glycoproteins that could be changing. There are changes could be happening in the heparan sulfate or the glycosaminoglycans. So when we're looking at changes in disease, it's the whole glycoconjugates that could be changing. Glycolipids could be changing, glycosphingolipids could be changing. So we can't just say glycoproteins. If we just concentrate on glycoproteins, we'll be able to understand what is happening with glycosylation. We'll have to look at the whole glycocalyx and understand the changes in glycosylation in all glycoconjugates within a cell in order to understand the disease and if glycosylation is important for that particular disease.
Understood. Yeah, it is a complex challenge, and which consists of many different elements, as you mentioned. These are not just glycoproteins. This could be glycolipids, and of course other molecules that kind of determine the phenotype, determine our response to diseases and response to specific therapeutics we use to treat them. And one more question that I would like to ask you is actually related to something that you mentioned a little bit early in this interview. So you said that at your institute, you are working a lot with the pharma companies, and we have seen essentially the boom of recombinant proteins used as therapeutic agents, and probably the most pronounced group of those therapeutics would be monoclonal antibodies. And of course, those monoclonal antibodies are also glycoproteins. They have this little glycan tree attached to them. And from what we know so far, it seems that this glycan tree is important for the activity of those antibodies. So I'm curious to know, actually, are there any ways to specifically modify those glycans on the surface of antibodies to change the therapeutic activity of those medicines?
So, of course, we worked with a number of companies on antibody characterizations, both small and very large companies. Antibody glycosylation is actually fairly simple compared to a lot of other proteins, but still challenging, because I think the challenge is to be able to accurately quantitate glycans on a particular antibody. We can always study the glycans and say, okay, the biantenary is the main structure. It has no galactose or one galactose. It has fucose or no fucose. But how accurately can we determine that? I think that's the challenging part, because quantitation of glycans still is a challenge in our field. To accurately quantitate glycans is still a challenge in our fields. Let's say if there are 15 or 10 to 15 glycans on a particular glycoproteins or antibody. We don't have, we should have all those glycans as standards to be able to quantitate those accurately. And those are challenging to have those standards purified in a purified form to be able to accurately quantitate glycans. Of course, glycoengineering is important for the antibody fields. Companies are changing the glycans using enzymes or the type of cell that the glycoprotein is being produced dictates the type of glycans that are present. Looking to see how we can quantitate these glycans accurately going forward. It's important to the field of glycobiology, not just for antibodies, but for other glycoproteins.
So, before I let you go, I would like to discuss the future with you, and I would like to get your insight into what do you expect to happen to the field of glycobiology in the next 10 years? Or maybe, let me rephrase it, what would you like to see happening in the next 10 years in this field?
I think it's a very exciting time to be in the glycobiology field. It's being recognized as an important field of biology in many aspects, with disease, with biofuels, with many aspects of life. So going forward, I think there are two areas that are important for our field. One is to be able to have the right tools to be able to study these structures. And I'm happy to report that funding agencies are showing a lot of interest in our field. NSF, Department of Energy, and NIH are all, I think, vested in this area. So with more funding in this field, I think new techniques will be developed so people will not be so scared of glycofield. And more people will come into this field and study the glycans, the role of glycans, of all types of glycoconjugates. So it will be important to basically have more people studying in this field, more people looking at these structures. But another important point is to be able to train the next generation of students and glycobiologists in this field. One of the ambitions of the Complex Carbohydrate Research Center in the last 30 years have been, and it's a mandate of us, to train the next generation. Our hands-on training courses in the last 30 years have trained more than 2,000 glycobiologists. They've come and taken our hands-on training courses. But we want to expand these. We want to make glycoanalytics or glycobiology accessible to a lot of people. We will continue to have these hands-on training courses, but I think it's important to train more and more students and faculty in this field for them to understand that glycans are important for their research. Any research that they're doing, glycans are important in that research. But the only way they're going to know is to be trained a little bit and to have an interest in glycobiology. So I'm happy to report that this year we'll have our courses online. They will be in August, and there will be a link in our website. They will be free this year because there is no hands-on. They will be just virtual training courses. But that's a good opportunity for a lot of people to get exposed to different tools that are available in glycobiology and learn about different researchers doing research on different aspects of glycoscience.
Perfect. Yeah, that sounds really fantastic, and we'll definitely make sure to put the link to those courses into our show notes so that more people who are listening to this podcast can actually get first experience in the world of glycobiology research. Perfect. Parastoo, I think this was an incredible episode, a very interesting interview, and I'm sure there are a lot of glycobiology enthusiasts, maybe mass spec enthusiasts, who would like to reach out to you and get in touch. So how can these people find you online?
They can reach me at my email, Azadi, Azadi, at uga.edu. They can also go to our website, ast.uga.edu, or just visit us at the Complex Carbohydrate Research Center. You can find out about the center, the different types of research going on at the center, and the different faculties. And there are developing techniques and new tools for all types of glycoscience that is going on at the center.
Perfect. That sounds fantastic. Thank you very much for being today with us. And I'm looking forward to talk to you again on this show, perhaps in a few years from now.
Thank you to you for setting up this podcast, and thank you for your listeners. It was very interesting. I look forward to speaking to you too soon as well.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to the Personalized Medicine podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Casts, Stitcher, iHeartRadio, and many, many more. Please rate us there and leave a comment. That helps us to grow and deliver the best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. And engage with us on social media, where we regularly share the news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook just by typing in Personalized Medicine Podcast, or use our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.